Good evening and welcome to the NFA North Campus Annual Science Research Symposium. Before we begin, I would like to take this time to express my gratitude to Ms. Oberle for her unwavering dedication to our students as they prepare their science research projects. Additionally, I would like to thank the exceptional faculty and staff of NFA North for supporting this event. I would also like to state my appreciation for all the members of the Scientific Review Committee who kindly accepted our invitation to support our student research projects. Thank you. Parents, guardians, and family members, I thank you for your support of your children and encouraging them to become critical thinkers. Last but not least, I would like to thank the brilliant and hardworking students who researched, planned, and are now presenting these exceptional projects. As I considered the theme of my remarks tonight, I reflected on the significance of science research, reasoning and my gratitude for the scientists who have greatly impacted our lives. And I wish our students well in their journey of becoming leaders in the field of science research. Every year, our NFA North Science Research students select, conduct, analyze, and report their research. Under the guidance of Ms. Oberle, mentors, advisors, teachers, and the Scientific Review Committee, these students were able to pose a question, develop an hypothesis, collect and interpret data, draw conclusions, and communicate their findings. It has become a true learning community. Parents, your unwavering dedication to our science research program has made students' success possible. Thank you again for your support. And now to our research students. You are able to enjoy this wonderful experience due to your scientific curiosity, which resulted in scientific inquiry. You have been encouraged by many individuals who have supported you to this culminating event, where you are able to demonstrate your ability to challenge, inquire, and discover the many layers of knowledge that you have gained. This level of support will not end at the conclusion of today's symposium. You will continue to promote your love and for learning for many years to come. I believe that everyone in our learning community will continue to promote our students' scientific enthusiasm and inquiry. I would like to challenge you to continue to push yourself by developing your research skills, review literature, engage in discussion, analyze data, write and rewrite, and yes, let's not forget to have fun. Again, thank you and congratulations. Have a good evening.
Greetings and welcome to the 9th Annual Science Research Symposium, our second virtual symposium at NFA North. My name is Kristin Oberle and I am the instructor of the Science Research Program. It has been such a challenging year for science research. Many of our students have faced so many obstacles with their intended research due to COVID restrictions and limited access to labs, lab equipment, and to mentors. It has also been extremely challenging to develop the community and team aspect that is so much a part of science research, because all students have been 100% virtual for the whole year. So I have not had the opportunity to work with students face-to-face this year, and they have not had the benefit of the face-to-face interaction with their classmates and team members. Practicing our research presentations has been online since September, and the classroom interaction and non-stop feedback from classmates is something that we all miss. But with all the odds, these students have still worked so hard on their own to structure a video presentation highlighting their research intentions. It is an honor and a privilege for me to work with such motivated young leaders who are committed not only to their own academic success, but also to the success of their classmates. We want to thank Ms. Holmes for putting together this fabulous video for us, Ms. Scars for creating the symposium display on the district website, and Mr. Dotto and Mr. Romano for helping me facilitate science research this year. Most of all, I would like to thank all of the science research students for their dedication to challenging college-level work, and to their parents who are there to support them through it all. And now the science research team invites you to enjoy the presentations that have been prepared for you today. Thank you so much for spending your time with us. We truly appreciate your support.
Hi, my name is Kira Sevilla, and this is my symposium presentation. My research is on the characteristics of music that affect audience emotions in movie scenes. Introduction. Music plays an important role in the background of movie scenes. Music in films helps create emotional responses, helps enhance the storytelling, and helps the audience build an emotional bond with the characters and the plot, all while the director's message is portrayed throughout the movie. Review of literature. Studies have shown that music can modulate perceptions, actions, and judgments in everyday situations. Among them, it helps shape emotional responses, creates a rhythm to scenes and segments, and comments on the action. Music is often crucial to the experience of a scene, and in some cases becomes as iconic as the movies themselves. An example would be Jaws. Objective and hypothesis. My objective is to analyze and compare characteristics of music used in scenes meant to evoke happy and sad emotions from audience members. I predict that loud, fast, and higher-pitched background music provokes happy emotions, while the opposite would be used to provoke sadder emotions. I believe that dominant instruments used for music in happy scenes would consist of flutes and drums, while music for sad scenes would consist of cellos and violins. Furthermore, I predict that happy music would have an average of 100 or more beats per minute, while sad music would average less than 100 beats per minute. Methodology. In order to reach my objective, I will review scene music from 5 different animated films for the following features: dominant instruments used, use of rising or falling scales, higher low pitches, and fast, slow, louder, quiet music. I plan to list these elements on a chart and take notes based on audience experiences. I will be reviewing the music in the following animated films: Beauty and the Beast, Moana, Coco, Tangled, and The Lion King. Expected results. I expect to see similar results for all scenes that use happy music. For example, the dominant instruments I expect to see in happier or upbeat music will most likely consist of instruments like drums and flutes. I expect to see string instruments like the violin and cellos to be used in all sad songs for movie scenes. Acknowledgement. I would like to thank Ms. Oberle for guiding me and helping me with my research, my peers for their comments and feedback, Dr. Sarah for being a mentor and for giving me access to his research, and Chris Mahoney, who also mentored me, and I would like to thank him for his ideas that he contributed as well.
Good morning. My name is Amanda Wade, and my research project is, does COVID-19 leave people with low oxygenation levels? For my introduction, COVID-19 is a virus that mainly attacks your respiratory system. Your lungs are the main organ in your respiratory system. However, COVID-19 also attacks many different systems in your body. One of these systems are the immune system. I, however, did not research the immune system being attacked for COVID because it did not pertain to my research. COVID-19 has been seen to last longer in different people and has also been seen to affect different people in many different ways. Some people have been hospitalized while other people have died, while other people just sit in their bed and wait for the 2 weeks to pass before they can get out of quarantine. The virus has been leaving many people with long-lasting issues, one of these issues being low breath output. Scientists have been looking for ways to prepare the lungs for COVID, but really haven't been looking for ways to help with the after effects. The main reason for this is because most of the after effects are not fatal. For my objective and hypothesis, the objective of my research is to find out if COVID-19 may leave people with long-lasting effects to their breathing. My hypothesis is those who have had COVID-19 will be left with lower oxygenation levels than those who have not. Materials. The materials that I will be using consist of a spirometer, disposable mouthpieces for the spirometer, a pulse oximeter, disinfectant wipes, cleaning fluid, and a container that I will place the spirometer in to disinfect it properly. Methodology. Before completing my experiment itself, a few things need to be done to make sure that there are no liabilities within this experiment. The first thing I will do is have volunteers fill out all necessary consent forms and volunteer forms. For minors, this will have to be filled out by their parent or legal guardian. Next, I will make sure that the forms are correctly filled out. Lastly, I will make sure that the forms are filled out correctly and file them in a binder or folder to keep organized. Methodology continued. Now onto the experiment itself. First, I will place my pulse oximeter on the volunteer's finger and record their data. Then I will explain how to use this barometer. Before placing on mouth, take in a few deep breaths. Take a deep breath in, blow into the spirometer as hard as you can. While doing this, you are trying to get the air or ball as high as possible. Next, I will have the volunteer complete a spirometry test 3 times and record all the data. Over the course of a week, I will have them complete this 1 time per day, so that will be 7 times altogether per volunteer. Acknowledgements. I would like to thank all the people who have supported me and helped me throughout my research. Kristin Oberle, Kimberly Poje, Dr. Victoria Romano, Dr. David Maria, and most of all my mother who have supported me throughout the whole entire process. This is my Works Cited page, and thank you for listening to my presentation. I hope you all have a good day.
Good morning, afternoon, evening. My name is Sean Zamanek, and I am a junior, and my current project is on equine behaviors. Equine behaviors. Can horses learn to trust again after years of abuse? Now, racehorses face severe abuse every day. Throughout their years on the racetrack, they have been worked to beyond exhaustion and face many forms of abuse, including starvation, performance-enhancing drugs, physical abuse, and medical neglect. Many of these highly abused animals are auctioned off to the highest bidder, or sold to processing plants for meat and glue, among other things. Those lucky enough to be auctioned to a new home now face forms of PTSD that many deem as untrainable. Now, my hypothesis. Throughout my research and hands-on work with retired racehorses and other previously abused horses at the Saddlebrook Rescue Farm, I hope to prove that it is not only possible to reteach these previously abused animals, but that the horses can learn to trust no matter what degree of previous abuse when you learn to interpret their body language using consistency and empathy. Now, my methodology for doing so. Now, empathy and interpretation of equine body language, I believe, are the center of this experiment. Once an abused racehorse has been identified and brought to the farm, they are paired with a babysitter horse, or horses, to form trust and bonding between the animals and to give them an equine guidance and as well as companionship. Routines for feeding, watering, pen and paddock cleaning is established and maintained. This is very important to establish trust. It is important to be aware that not only is the horse's body language, but yours as well, as an act of dominance can destroy trust, building and bonding. Never approach the horse, let it come to you. Now, usually use a gentle tone, relaxed body language, and empathetic eyes when building trust. Always be present and aware. Equine behavior, like other animals, can quickly change. Tacked ears, high head, gritting teeth, head shake, body quiver, change in breath, wide eyes, are all subtle precursors to bad behaviors which can be avoided. Do not punish a behavior. This can do more damage than good. Walk away from the situation and begin again at the next scheduled time. Reward good behavior. Treats, praise, rub downs, and caressing are great forms of rewards. You use multiple rewards to signify different things, and you treat good behavior, which can help to improve their behavior. Now, the 7 steps to success. The first one would be bonding. Touching is important, but so is building trust. This often starts with babysitter horses, but gradually builds to human trust. Number 2, consistency. Start with feeding, watering, and cleaning at a scheduled time each day. Then add visiting, sitting out without expectation, or even reading to the animal until it approaches you. Never force contact. Number 3, gentle, non-threatening approach. Find the sweet spot. Now, the best way that I can use to describe this is if you ever had a long day and someone just comes over and scratches your back or rubs your shoulders or something like that, and before you tell them to stop, they stop, and you're like, hey, come on, really? It's that sort of thing where you don't want them to stop. It's that sort of thing where they come back wanting more. Number 4, identifying their fears and phobias. Find a way to build tolerance to the phobias. Now, finding the fears and phobias can be challenging, but with enough work and enough time, you can manage it. Now, number 5, establishing boundaries and setting goals. Rewards, whether praise or treats, are helpful in keeping training moving in the right direction. As I've said, negative or forced behavior is never a good thing. It always causes damage. Number 6, be aware of body language, yours and theirs. Low head is relaxed, high head is stressed. And finally, number 7, never leave a sticky situation alone. Keep at it. Every animal has its own pace. In conclusion, these beautiful creatures can learn to trust humans again, giving them a purpose outside the boundaries of the paddocks. Consistency, patience, and empathy help to build trust where before there was fear. Over time, these horses can be retrained as trusted teaching animals or trail guides, while others are trained as therapy animals in programs for mental and physical disabilities. Many of the horses I've been blessed to work with were once abused racehorses who have now become loving, trusting friends who live amazing lives. Like any animal, these horses still occasionally face phobias and reoccurring fears that make them fearful, but never again will they face the abuse of yesteryear. They now get to bask in the sunlit fields while helping people who need them as much as they need us. For my references, I would like to thank Ms. Oberle, and I would also like to thank Helen King at Saddlebrook Rescue Farm for allowing me to do this experiment with her. Thank you. Any questions?
Hello, Go Back family and parents. My name is Cyrus Boykin, and I'm a junior at NFA North campus, and my research involved investigating the effects of radio frequency electromagnetic field radiation on plant growth. So, radio frequency electromagnetic field radiation are everywhere in our daily lives, whereas it empowers most of our technology, such as smartphones, computer tablets, smart applications on radio, and has several applications such as for the field, commercial, and business and entertainment, along with countless others. And basically, technology is so much plays a huge role in our lives, and we constantly use it day by day. And that whereas we are constantly exposed to our RF radiation on a daily basis for 24/7, and such would be more the case as the new technology such as 5G and countless others develop and roll out. And as we be more and more reliant on technology and use it much and much more. And such is concerning as exposure has been reported that have some effects on living organisms in the environment. So such living organisms include but not limited to humans, birds, plants, and rats, and such have some cellular molecular changes, along with increased risk for diseases, as I will show in the next two slides. And with that being said, a few studies have been done investigating the effects of plants from higher frequencies of RF radiation. And I, as this is concerned, I investigating the effects from the exposure of RF radiation on plants is important as plants have similar cell structure as humans and animals, and observing effects from exposure can add question and open door to how that can how that can extrapolate to human health and animal health. So what are electromagnetic fields and radiation? They are a combination of electric and magnetic fields which are generated through an electric charge, and essentially light where there's divided to different forms of light is that characterized in the electromagnetic spectrum. So most of it, our technology uses radio and microwaves, and the electromagnetic waves would be characterized here as you see here, but carry packets of energy and stuff, and with a higher frequency. And new technology would use higher frequencies. With higher frequency, we would have increased energy, but though it would have smart wavelength, which would mean it would travel less in distances. Would mean it would use, and such application would be used for 5G for the sites. It would need more infrastructure into building and for more reliability into it, and for its efficiency for 5G, which would mean it will be more exposure from our RF radiation, particularly higher levels. Some reported effects includes in cellular molecular. So EMF exposure has been reported to cause an accumulation of an inflammatory molecule called peroxynitrate, which is known for having DNA damage and inflammatory responses in humans, along with decreased mitochondria and cellular efficiency in transporting nutrients and oxygen in the body, which also can have increased risk for oxidative stress and cell death with peroxidation and increase in some diseases, as shown on the right, with such as electromagnetic hypersensitivity, which is characterized as headaches, brain fog, insomnia, and some pain, along with increased risk for CVD, diabetes, and some cancers, along with reduced sperm reduction in men, and increased infertility in women, along with increased risk for neurological disorders such as autism and the disruption of the blood-brain barrier. And this is more concerned with little kids and for infants, as they have small brains and smaller skulls, meaning our EMF radiation can easily travel through the brain and have them more exposed and having the brain more vulnerable to neurological disorder, especially from long-term exposure. And then on plants is changes in morphology and increase in sensitivity in some plants, along with some effects in some animals, such as some killing some bees and birds, and also how it alters their magnetic reception, so for navigation, with increased stress responses and reduced ability in reproduction in some animals, such as in rats and yeast. And then lead me to the question of how would exposure from RF EMF radiation affect plants, and how would it affect extrapolate to other living organisms. And my hypothesis, um, the plants that will use that expose RF EMF radiation will show stunted growth rate and morphological changes. So here are some of the materials that I've been using for my methodology as reported, and this is for my experiment. As I will show my procedure. Um, I will use 40 plants, and then when I will use that, I will have half of the plants was 20 inside a 30 cage and with the router, so it can be exposed. But that will be classified as irradiated. The plants will be irradiated with that have another 20 non-radiated. But as shown in the picture, as the diagram, that's my setup here. And then I'll just let the plants grow with 3 to 6 weeks. Within 3 to 6 weeks, then the varying plants I'll be using, and then I'll record the temperature recordings from the digital thermostat I'll be using for at least three days a week, and record record measurement recordings on a notebook. And within 3 weeks, I'll measure their morphology with a ruler and record the measurements. And then proceed to do the same thing in week 6. And then lastly, I'll compare all data from initial to final measurements. And then how does it help in the scientific knowledge, and how would this be helpful for society? Let me just open the put more of that understanding, so we can take more precautionary measures into EMF radiation, and also focus more on the ethics on technology uses, and again take more precautionary measures into using it and from being exposed, and not only protecting for us living for us and for other living organisms, but also for the environment. Yeah. And my summary main objective of research is to determine effects from RF EMF radiation from plant growth and morphology, and then from the observed effect, hypothesize on the effects on how that would extrapolate to living organisms such as us humans, and actually contribute more on the understanding of the impacts of RF EMF radiation to both the scientific and general community, and then focusing more on the safety and ethics, improving the ethics of the technology. And then my acknowledgements are my parents and family for motivating me to keep moving on. And I'd like to thank Ms. Oberle, my science research instructor, for helping me giving me tips and suggestions for my research plan. And also I'd like to thank my mentor, Mr. Toplick, for suggesting that I focus on investigating the effects from exposure on plants, not only for simplicity but also for reliability. And also I'd like to thank my teachers, my other teachers from my classes, and classmates and peers, and for their support and questions and answers. And lastly, I'll end a quote from Isaac Newton, and that is, "If I have seen further than others, it is by standing upon the shoulders of giants." And here's my sources, and that is it. Take care.
Hello, my name is Daniel Rigo, and my topic is the effect of the intestinal microbiota on antibiotic and probiotic metabolism. The intestinal microbiota is also called the gut flora or gut microbiota, and they are microorganisms, specifically bacteria, that live in the digestive tracts of animals and humans. There's many factors that can alter intestinal bacteria composition, such as antibiotics during antibiotic therapy due to an infection, your age. As you get older, your intestinal microbiota start to die out, or the usual species that's supposed to be there to help you break down certain foods and drugs is not there, and your diet could also affect that. These are all very important because the intestinal microbiota is also called a secret hidden organ that helps us break down our food and drugs to help aid in the intestinal absorption. Excuse me.
Also included in this research is the idea of probiotics, and probiotics are basically a bacteria or yeast that helps encourage the replenishment of intestinal bacteria composition when it undergoes an imbalance. And such imbalance is called dysbiosis, where there is a microbial, basically there's not enough of a certain type of species for the breakdown of a certain type of food or a drug to be allow for total absorption into the bloodstream. So, two questions is how does that I will answer in this presentation is how does the antibiotics affect your intestinal microbiota, and how does the antibiotics and probiotics interact with each other in the human intestine? That's basically what my research is about. So, reviewing literature, many articles have said that intestinal microbiota contains tens of trillions of microorganisms and has been known to have at least 1,000 different species of known bacteria with more than 3 million genes, which is 150 times more than human genes. And such prevalence of bacterial species in the intestines have a commensal relationship, which means they aid in the breakdown. They aid with the human body. They help the human body break down nutrients prior to intestinal absorption, which is why this is such important. Everybody has a different type of community of bacteria in their intestines, and it's called an enterotype. Dysbiosis, which is the imbalance of the intestinal microbiota, has been known to cause a variety of cardiovascular diseases, obesity, gastritis, and alter nutrient drug metabolism. And drug metabolism due to intestinal dysbiosis will likely alter the way that we break down certain medications, such as antibiotics or Advil or any other type of drug, and food including. So the objective and hypothesis. So the objectives is basically to quantify the microbiota, screen different drug classes, in this case antibiotic classes, to determine trends in microbiota-mediated drug metabolism, screen, and as well as probiotics that would usually be administered during antibiotic therapy, to also determine trends in probiotic-mediated drug metabolism, and to screen represented microbiota species. The hypothesis of this, the hypothesis is that drugs are metabolized in the intestinal microbiota before intestinal absorption, and that the probiotics metabolize the antibiotics before intestinal absorption. Overall, my goal is to see the different products of culture gut bacteria and analyze it to see whether it is beneficial or toxic. Also to see the interaction between probiotic and antibiotics in a gut-stimulated environmental culture. So some materials include, for a common culture with bacteria, include petri dishes, pipettes, agar-agar, water, gloves, microscope, test tubes, stoppers. The LCMS, which is a liquid chromatography mass spectrometry. It's a machine that helps me see the breakdown of metabolites or products that the bacteria is broken down with regarding to adding antibiotics or probiotics. And in this case, we would be using azithromycin, which is a broad spectrum antibiotic that's usually prescribed by doctors a lot, and amoxicillin, which is also a commonly used antibiotic. And for our probiotics that we'd be using, we're going to be using S. boulardii, which is a type of yeast, and the different bacteria that we'd be using is Peptostreptococcus sp, Bacterium sp, and Lactobacillus and Clostridium sp. So the methodology, first of all, we develop a microbiota representative bacterial library to understand what types of bacteria, what's their functions in the human body, and to advent a next generation of DNA sequencing to allow a more sophisticated analysis and sampling of these complex bacteria colonies, which brings me to the selecting and analyzing a subset of GI microbial strains with individually and heterogeneous colonies. Then we will also select representative drug classes, which will allow us to specifically study its function of the drug class, its contents, and how it's usually broken down in the body. And then we would analyze a subset of drug structural motifs, yet again, study the different antibiotics, its formula, and again, how it's broken down in the body. And then we would culture the bacteria in media. We would culture the bacteria in the media, let it grow for a while, add the antibiotic to some cultures, which is our control. And then for the probiotic aspect of this research, we would add, for one set of cultures, we would add the antibiotics and the probiotics. And then in the other cultures, we would add the probiotics and then the antibiotics to see what would occur during probiotic therapy, during an antibiotic therapeutic time period. Then we would use we would develop a liquid chromatography mass spectrometry method to quantify the parent drug and metabolites. For analysis, we would be analyzing the concentration of antibiotics, the products of the metabolism, as a result of adding the antibiotics and probiotics, and then we would determine the interaction between probiotics and antibiotics and its probable metabolic products. This will all be done by using a liquid chromatography mass spectrometry method to determine how much antibiotics is left in the culture, what type of metabolites are in the cultures, to see what type of reaction would occur if it was in the human body, if it was absorbed in the bloodstream, which would determine if it was a beneficial, toxic, or just neutral reaction as a result. And then we would compare all these additional metabolites to see how it would interact in the human body. The benefits of this research would help us create a better understanding of the intestinal microbiota and its role in antibiotic and probiotic metabolism. It would also clarify how each of the enterotypes metabolize antibiotics and probiotics, which will help improve antibiotic formulas and treatments, improve probiotic formulas, and improve treatments for people suffering with intestinal dysbiosis-related diseases such as cardiovascular disease, gastritis, obesity, and etc. I would like to thank my current mentor right now, Major Preston J. Dilly from West Point, and my former mentor, Major Aaron E. Milner from West Point as well, who would be helping me with my experiment at West Point. I'd also like to thank Ms. Oberle for organizing and helping me with my research, and my peers for giving me ideas. This is my work cited. Thank you for watching my presentation.
Hello, my name is Zachary Dawn, and today I'll be presenting my research on microplastic concentrations in the Hudson River. What are microplastics? Microplastics are pieces of plastic that are less than 5 millimeters in size. They occur from either the production of small pieces of plastic or from the breakdown of larger pieces of plastic. The photo on the right shows 5 different classifications for microplastic. It shows fiber, film, pellet, foam, and fragments. Microplastics are one of the largest forms of pollution in the waters today, and the effects of microplastics on humans are still not known. However, it is estimated that we eat about a credit card's worth of microplastics in a week. And there are many different methodologies to collect and sample microplastics, which is an issue when trying to develop legislation to correct microplastic pollution. My problem statement: like I said, microplastic studies do not have a common methodology to replicate for new studies. Because of this, we can't compare and contrast microplastic studies from different locations, and the methodology that I developed needs to be tested before I use it in further research. In addition, there's just a general overall need for more microplastic research. It's an under-researched field, and the more research that comes out, the more that we learn about how dire it is that we need to correct this problem. The development of my methodology: there's no standard methodology for microplastic collection and sampling. As I said, because of this, I couldn't just take a study and recomplete it. So me and my mentor had to take parts of other experiments and combine them to make the simplest methodology. I like to break down the microplastic methodology into 3 different steps. So there's the collection, filtration, and the observation. For the collection, there's 2 different main methods of collection, the samples from the river. There's using a plankton net and a glass jar. A plankton net would just simply be dragging a plankton net along with a boat in the river and taking the sample, and it leaves you with just anything solid that couldn't go through the net. And the glass jar method is more simple. You just simply take a glass jar and dip it in the side of the river, and then you would filter out the water. And then there's the methods of filtration. So if you use the plankton net, you don't need to filter the sample much because the plankton net has already filtered the water out. However, if you use the glass jar method, you need to use vacuum filtration or any other type of filtration method to remove the water from the sample. In addition, some methodologies use chemicals to remove organic material from the sample, which makes the observation much easier. And then that leads to the last step, the different methods of observation. The two methods to observe microplastics under microscopes were using compound microscopes or FTIR microscopes. I had access to compound microscopes. FTIR microscopes are much harder to come by. However, these could chemically analyze the sample and figure out using a computer program what is microplastic and what is not. My methodology, first I used a 1 liter triple rinse glass jar and I took a sample of water from the edge of the Hudson River. I just filled it to the top and sealed the jar, and that was all I had to do for the collection. Then I took that sample back to school with the school lab and I placed it to the side as I set up the filtration setup. The filtration setup consisted of gridded filter paper, a clamp, and the water would run through a funnel. The funnel was vacuum pumped with to allow the water to go faster. It was vacuum pumped manually with a hand pump. I would also use a squeeze water bottle to squeeze a little bit of water onto the filter every so often to help the filter process just go a little bit smoother. Then I would place the wet filter onto a slide and I would observe it under a microscope at 45x magnification, and I would characterize the microplastics using physical characteristics, and I would record it. The goals for my research, I want to test the efficiency and effectiveness of my methodology. Like I said, I had to combine different methodologies from different experiments into my methodology to do what works best with me and what materials I have access to. So I had to make sure that it was correct before I continue on with my research. In addition, I wanted to have practice sampling and separating microplastics before I go on to more research that is more important, I would say. And I want to use the knowledge that I gathered to form my final research question and experiment that I'm going to be working on over this summer. Some of the issues I faced while conducting my experiment, analyzing the samples correctly under the microscope was one of the biggest issues that I faced. Human mistakes is the largest variable and error in this experiment because it is difficult to tell if something is a microplastic or if it is, for example, a leaf or a fish egg. In addition, I didn't have all the access to the materials that I needed. The gridded filter paper that I had was not the correct size, and it did not filter all the microplastics. In addition, there's also the possibility for air contamination. There's microplastics in the air as well as in the water, and my sample was exposed to the air for a good amount of time. There's also the possibility that when I do this methodology again, that there's too many microplastics to count, and that I didn't complete the experiment correctly and I didn't follow all the steps because it is a complex experiment. Moving forward, the current study that I have is not feasible with the materials that I have access to. If I want to have a study that has accurate data so that I could use it to present to scientists, and the study that I have is not allowing me to do that. Because of that, I'm going to be working with Dr. Klaus of Marist College and one of his students to assist them with their sediment microplastic study, viewing sediment samples from Long Beach Island. This is similar to my research. I did river microplastics. They're doing sediment microplastics, but it's a similar methodology. However, they have access to the materials that I need to have it more accurate. And I'm going to continue doing editorial research on microplastics in the Hudson because it's something that I'm just very interested in now that I've read about it for the past 2 years. And I think that more research should be done. And there is some research being done, especially down by the city. So I'm glad to see that. Some of my acknowledgements, I want to thank Ms. Oberle for always guiding me in the right direction this year. It was a difficult year for everybody, and she was always there for me, giving me pointers and giving me suggestions when I needed it. Also to my classmates for being there with me and giving me suggestions on presentations and whenever I needed it in class. Mr. Caldwell, the Crusade Creek Watershed Alliance, for giving me pointers and pointing me in the right direction to meet professionals in the field. Maya Nimestro of the Department of Environmental Conservation for giving me the advice that I need and mentoring me to make my methodology and combine the different aspects into one. And Dr. Claus of Marist College for working with me moving forward with the sediment samples in Long Beach Island. These are some of the citations that I use for this presentation. And thank you.
Good afternoon. My name is Shema Herzala, and my topic is about the effective exercise on the memory of early-stage dementia and Alzheimer patients. So what is dementia and Alzheimer's disease? Dementia is a long-term brain disorder causing personality changes and impaired memory, reasoning, and social function. Dementia manifests as a set of related symptoms, which usually surface when the brain is damaged by injury or disease. It is also a group of conditions, and one of those including is the Alzheimer's disease. So how is dementia related to Alzheimer's? Alzheimer's disease is the most common cause of dementia. Although not all causes of Alzheimer's disease are known, experts do know that a small percentage are related to mutations of 3 genes, which can be passed down from parent to child. Alzheimer's disease is a disease that usually starts slowly and progressively worsens. As the disease advances, symptoms can include problems with language, disorientation, mood swings, loss of motivation, self-neglect, and behavioral issues. This can also cause atrophy, and atrophy is a process of degeneration of muscle tissues of the brain. And atrophy is a shrinkage of the brain, and this could lead to death, and it is shown that exercise improves the intellectual capacity of the brain. The main purpose of this project research is to determine specific treatment using physical activities that could help improve or slow down the process of developing memory loss diseases, such as Alzheimer's or dementia, and to find ways to improve memory and quality of life for those who are currently suffering or have a chance of suffering from this disease in the future, and also to provide proof that a physical activity has an effect on Alzheimer's disease and what are its benefits. So the methodology for this research project was a systematic review, and in this review I was able to gather information from about 17 articles and have been reviewed. I then placed the data on the chart to compare each article by the age group, gender, physical activity, results, sample size, and whether the results were effective or not effective. As you can see here in my chart, I have separated them by article, age, gender, type of physical activity, results, and sample size. Each of these categories are very important as it will give us a higher chance of determining whether physical activities does indeed work for everyone or just certain groups of people. I did the same thing here. This is a continuation to it, as well as here. So the results, as you can see in the charts, it's shown physical evidence that proves that physical activities and exercise is a common preventable risk factor for developing memory loss diseases, including Alzheimer's, and it can have a positive effect on Alzheimer's. It does not include or pertain any harmful chemicals, such as the medications that some take to reduce the effects of Alzheimer's it has on the person who is suffering from it. Physical activities is showing to be an alternative, non-harmful solution to Alzheimer's disease, as it does not require any chemical compound intake to the person's body. In this article, is proof that fewer side effects than medications. Physical activities did not have many cons, and it had many pros, and it proof that it helped the cortical thickness to thicken. For this article, they only use females because it is shown in evidentially that females are the most vulnerable to this disease. And also, in this article, they only use males because they wanted to see how come women develop the disease and are more vulnerable to it instead of males. And they wanted to test with male hormones to compare them to the females. Circulation increases the blood flow, rising the oxygen levels in the body, leading it to function more effectively. Exercise in general is a very healthy choice to lean towards as it will reduce your risk of developing any type of disease by a 50% chance, especially for Alzheimer's. Another huge component that is useful to prevent Alzheimer's is social engagement. Alzheimer's disease requires social engagement. The more you socialize with others and do brain-powering activities, the less likely you will develop Alzheimer's disease. However, although there is physical proof that show that Alzheimer's does improve memory, and more studies are still needed to provide more answers and to take different routes on this topic. We come to conclusion that for this research project that the current findings show that exercise and physical activity had a significant impact on memory loss diseases, as it improves and lowers the risk of developing such diseases. Exercise not only diminishes the risk of developing Alzheimer's disease or any memory loss disease, but it also reduces the risk of developing cardiovascular disease, Parkinson's disease, depression, and diabetes for those who may be already suffering from Alzheimer's or may be onto the route of developing it later on. Scientists suggest that aerobic exercise is the most effective way of not developing Alzheimer's disease, and a person should exercise for about 30 minutes daily. As you can see in this picture, this is atrophy. In A, it is the first stage of atrophy. And in C is like when it starts to degenerate. And then B is the mild. And then D is when the atrophy is completely has taken over the brain. And then mostly in these cases, that is what usually occurs. I would like to give a huge thank you to Dr. Claire Thomas Bino, who have helped me with this project and helped me perfect it. And I want to give also Dr. Victoria Mano, who have helped me include these articles in my research project. And my teacher, Miss Kristin Oberle, because without her, I wouldn't have been able to have done this project. These are my citations for this project. And thank you very much.
Hello, my name is Darcy Guerra, and my topic is a systematic review of the effect of the type of bilingualism on the cognitive functions in children. For the systematic review, all results concluded bilinguals having some advantages over monolinguals, from better executive control with attention and task switching capacities, which can contribute to better test results, as well as having connections and correlations to age-related variables. An article by Solomoni in 2018 increased interest in the different types of bilingualism and their varying effects on advantages. The objective of this systematic review was to help create focus on different types of bilingualism based on acquisition and fluency. The question that was being researched had to do with types of bilingualism and advantages that are able to be obtained from the varying degrees. The hypothesis stated in this study was if a bilingual is more fluent in a second language and learned it at a young age, then they will have faster results on the cognitive tests compared to other types of bilinguals. The tests used were neuropsychological tests that measure executive function by inhibiting cognitive interference. Executive functions are the subconscious process of using developed skills to obtain a given goal. The types of bilingualisms that were studied were divided into three categories: early, late, and passive, with early being divided into two subcategories: simultaneous and successive. These types differ in age and the way of acquisition. For the methodology, the first step was to formulate a question, the second to conduct research and collect relevant literature. The third step is to organize all literature and noting constant variables that could have an effect on outcomes and conclusions stated. For this specific research, variables such as age, gender, population size were taken into account, as well as types of bilingualism, if applicable, were determined. The fourth step was to analyze data and combine results for overall review based on the patterns that were noted. For overall data analysis, this systematic review started off with 38 articles to be examined, but were slowly filtered out due to many obstacles. The results of the experiments were noted in the last column of a data table, specifically conclusions of results of tests that were given. In this systematic review, a separate and additional table was created to identify and explain the different tests and throughout the experiments in this research. This table shows some of the literature used and exemplifies how it was divided based on the variables, and if applicable, the type of bilingualism was determined if that's sufficient information. The last column having the notable conclusions and results that were drawn from the experiments and their tests. This is a data table that collectively shows and exemplifies the different tests that were used and their correlations and measurements with the executive functioning. In this systematic review, many different age groups were examined, giving widespread of data, allowing for a thorough examination of the variables. Types of bilingualism were determined through information towards acquisition and noted if applicable. The last column helped to conduct and conclude overall patterns seen in the advantages among the variables. The most significant study having similar outline of this specific review had concluded more balanced and younger acquisition ages having better performance on the test. Conclusions drawn from this review: overall, there are advantages in bilingualism, especially early acquired and balanced bilinguals. The hypothesis in this research was proven to be correct and supported some studies that have been able to prove the differences and advantages in bilingual types, showing faster improvements in tests and overall time due to the development in cognitive functions, and advantage toward attention control due to the language switching skills. For future research, an experiment was planned with the objective to find differences and advantages of different types of bilingualisms that were presented in children. A planned experiment on the focus question of this systematic review would have participants fill out consent forms, questionnaires, and complete the stroke test, which would have participants be limited due to certain obstacles in the stroke test, which was decided for this experiment. For this experiment's methodology, the proper forms would be sent out and results would be recorded to have participants divided into groups based on bilingualism and have results analyzed in those groups. These are the sample questions for the questionnaire that would obtain the information of language acquisition that would have bilingual types to be determined. For the expected outcomes of this experiment, the goal is to view significant differences in bilingualism and encourage and promote language learning at a younger age. Expectations would be the same as a systematic review hypothesis. I would like to thank my mentors, Dr. Jeffrey Room and Dr. Kathleen Gaher, for their advice, knowledge, and support throughout this process, as well as my fellow classmates from their feedback and support, as well as my science research teacher, Ms. Oberle, for guiding me through this entire process. Thank you.
Hi, my name is Arnaz Riza, and my presentation is on a comparative study between White Claw and other bacteriophages of the same cluster. To begin, if one were to take a walk outside to observe nature, the thousands of tiny microorganisms thriving beneath their feet would remain unnoticed. Many of these microorganisms are bacteria, but at least 10 times as many would be their biopredators called bacteriophages. There's an estimation of 10 to the 31 phages on Earth, and each of them play a vital role in their environment. Scientists are mainly interested in phages because they help in understanding fundamental molecular biology. Although genomic analysis of phages have been available since the 1990s, DNA sequencing technology has advanced rapidly since then. Thus, using both phage isolation and genomic analysis will help further our understanding of phage viral diversity and even give us insights into bacteria/phage evolution. Most recently, phages have been studied for their potential to act as therapeutic agents. This is because the rise in the misuse of antibiotics has led to an increase in antibiotic-resistant bacteria, and so phages can present an alternative because they have bacteria-killing ability. The problem is there remains patchy knowledge on phage biology. Although several thousand unique bacteriophages have been discovered that infect various bacterial hosts, new phages are continually being found that are remarkably different genetically from any previously discovered. The objective of this study was to isolate and annotate a phage named Phagus. However, due to the COVID-19 pandemic, I did isolate Phagus from the soil, but I annotated a different phage named White Claw. So in following my objective, I asked the following questions. How much can White Claw as a Gardnerella phage be further characterized through bioinformatic analysis? Does it share any differences or similarities with phages of the same cluster, and what do these comparisons reveal? Understanding the relationship between White Claw and other phages in the same cluster was the key objective. So briefly touching on isolation, it really involves extracting phages from the soil, removing bacteria that are potentially dangerous, seeding the filtered sample, and incubating the cultures. Purification is ensuring the phage sample contains one kind of phage, and amplification involves creating a larger volume of the phage. Moving on to genome annotation, it's all about predicting the functions and features coded within a phage genome. Bioinformatic analysis with the help of computer programs helps to determine patterns by sequencing gene similarities to other known proteins. And it's important to note that annotation strictly involves relying on previous phage annotations and comparisons. This is mainly for two reasons. It's easier to predict the phage's genes, and second, building from previous annotations, there's less likelihood of omitting genes or functions that should be included in the annotation. All the genes were recorded in a program called PICHON, and two other programs, GeneMark and Glimmer, helped to analyze the phages. For example, if there were 41,692 base pairs of DNA, it would be computed as predicted 67 protein coding genes. White Claw has a total of 76 genes. Now the first step of annotation is to look for evidence of a gene. We do that by asking 4 questions. Do both Glimmer and GeneMark call it a gene? Is there coding potential? Do multiple similar phages have high alignment? Is there significant overlap with other genes? The second step is looking for the best start site. We ask 6 questions. Does StartRater program support this start site? Does this start site contain all coding potential? Do both GeneMark and Glimmer call this start site? Is this the longest open reading frame without significant overlap? Is this the best start site according to NCBI and Phages DB Blast? And does this start site have the highest ribosome binding site score? StartRater is a program that creates a report showing where start site calls have been made for similar genes in other genomes, and the higher percentage of phages in the same fam calling the same start site, the better the evidence. NCBI and Phages DB Blast programs are used to determine if other phages have called the start site for that gene. The third step of annotation is assigning functions to the genes. The majority of genes will not have an assigned function to the predicted amino acid sequence because sequences are too different from proteins that have been biochemically characterized. In addition, since annotation involves comparing already annotated phages to newly isolated ones, genes are not assigned functions without concrete evidence. So the 6 questions we ask are: does NCBI support a function? Does Phages DB Blast support a function? Is there HH-Pred data that supports a function? Is there synteny? What percentage of annotated genes in the same fam have this function? And is this predicted to be a transmembrane domain protein? In answering all 15 questions, White Claw 76 genes were annotated for the best start sites, 43 functions were assigned, and genes 35 and 42 were removed. In the 4 pictures below, we see that the 76 genes were annotated correctly. NFK means no known function, so the questions were not able to be answered properly to assign a function. We move on to some figures. We have 2 phages here, Soil Assassin and White Claw, and the background color matters here. The purple indicates most similarity and the white indicates no similarity. So in figure 2, we see purple in the background between genes 1 through 11, which show that the genes are roughly the same length and the same function, and we could also see through the picture that they are the same length. Figure 3, we're still seeing the same 2 phages, but instead we see white in the background between genes 29 to 34, so the genes are also different in length, and gene 30 for White Claw is an excise, but Soil Assassin gene 30 appears to be no known function. In figure 4, we have a different phage, which is Haley 23, and we see purple in the background. So if we notice the same genes we did in figure 3, genes 29 to 34, Haley 23 and White Claw are similar, but this was not the case in Soil Assassin. Additionally, gene 30 for Haley 23 is an excise, just like in White Claw. So while White Claw did not share similarity with soil assassin, between genes 29 to 34, it does share similarity with Haley 23. In conclusion, White Claw was successfully annotated and submitted to GenBank. It was seen that White Claw shares similarities in different areas / genes with phages of the same cluster, and phages that are placed in the same cluster have more than 50% genetic identity. So it does make sense why White Claw was placed in cluster CZ2, because White Claw exhibits tons of similarity with phages in the cluster CZ2, even though there are a few differences such as gene length and function. Future research should involve studying and testing more phages like White Claw for unique genomic properties that may highlight phages as potential alternative treatments for bacterial infections. Though one phage may not work at treating a bacterial infection, there are thousands of phages in the world that can be isolated and investigated. I'd like to thank my mentors, Dr. Sukarna Bala and Dr. Evan Merkhofer, for their guidance and opportunity to learn so much about bacteriophages and genes in general through the C-Phages program. I appreciate my partner, Gabriella, as we both had to accommodate to COVID-19 and work on White Claw's annotation together. And I'm also thankful for my teacher, Ms. Oberle, my peers and classmates, as their encouragement was beyond helpful. My references, and thank you for listening.